Um, well, the family of psychopharmacology has been embracing our distinguished chairpersons as a member of our family. Thank you to both of you for being here. And it's a privilege for me to be in the same session with the honorary president. Today I will be speaking about neuroinflammation. In particular, I will try and have a look into Neuron Microglia Crosstalk. We are all quite familiar with the different dimensions of the mental diseases in link with the pathologies of the brain. As we keep speaking throughout the Congress, social interaction, lifestyle, genetics, environmental factors, pathophysiology mechanisms, and neurocognitive models are really the parts of a very big puzzle that should be involved in our assessment of mental diseases. So we really have a wide variety of factors that are interlinked to each other, which I would assume would make a pharmacological treatment quite challenging, particularly in expecting the desired outcomes. I'm a theoretician as a pharmacologist, as you know. Of course, you as clinicians would have a better understanding in your observations. Clinical response is not always at the desired level. Most of the time, the picture goes on quite complex, presenting with relapses most of the time. This visual I like to use a lot because, as a matter of fact, we have quite a high variety of factors. In such an environment, we try and look at the issues from our own perspective. Therefore, it really matters where we're looking, how we're looking, because this would make an implication over the final outcome. This is my analogy to neuroinflammation. There are many events and actions involved in the process which might contribute or counteract our assessment. So the devil is really in the details, I need to say. And neuroinflammation is the very area that would explain the situation because there are many details that would make our jobs harder and harder every other day. Today I will be concentrating on microglia. Microglia cell is really a very important cell that is interlinked with the rest of the network. In 1880, it was for the first time characterized by French Nissel, and after that we have realized that it has really important functions in the neurological system. It constitutes about 10 to 15% of the cells found within the brain. Actually, I am not going to speak about history of everything, but this is an important turning point. Well, to take a closer look at the glial cells, microglia or astroglial cells are quite heterogeneous indeed. It's not about morphological variances, but at the same time, functional differences are also involved within this network. Therefore, we need to speak about, based on this diversity, of course, we need to speak about a new potential therapeutic approach, which at the same time would be quite challenging because of this feature of the area. As for the neuronal cells, neuron, microglia, astrocytes, oligodendrocytes, and oligodendrocyte progenitor cells are together involved in a very intense communication and in very close link with each other. So the functions and pathologies of these cells are just supporting each other in this very complicated network. In this respect, of course, astrocytes would deserve just a minute of speaking because, in terms of psychological aspect, they are involved in the anomalies at the neurotransmitter level. These cells are quite definitive, therefore, in the neurobiology of the depression, astrocytes have a big contribution. On the other hand, oligodendrocytes are important in pro-inflammatory cytokine release, to the myelin sheath formation. So in a very broad perspective, they would contribute to the neurobiology of depression, potentially. Whereas microglia at the same time will have not only in inflammation, but at the same time in synaptic and neuronal plasticity, has also a very big contribution to play there. Therefore, the amount of the neurotransmitters regulation also is involved as a part of the task of microglia. Therefore, microglia cells will have a contribution to the depression neurobiology as well. Let's take a look at these cells vis-a-vis the development of the nervous system, particularly progenitor cells, their migration and growth, synaptogenesis, specification, and healthy execution of neuronal functions at all levels can be provided with the control of the glial cells. Therefore, between the neurons and the glial cells, there is a very intense communication, as we know today. For the last 1.5 years, we have been speaking about this new term, neuroimmunometabolic sensors. Well, as a matter of fact, metabolism, immune system, and central system functions should be assessed altogether according to this terminology. In a healthy brain, most of the time we concentrate on the neurons with lipid, amino acid, and glucose metabolism. This metabolism is controlled mostly by the glia, astrocyte, and oligodendrocyte cells. Of course, when there is a dysfunction in any of these cells, as much as neurons, the pathologies of these cells would matter as well, because at the end of the day, altogether, with the collaboration and between all these cells, neurodegenerative or psychiatric disease may start. And now we are able to speak about this interaction at a broader sense. In this respect, astrocytes, microglia, and neuronal cells at the resting phase are activated by some stimuli. As a result, in the part that is reflected on neuron, we come up with neuroinflammation. In this respect, we need to look at the astrocytes as a part of their involvement in developmental biology. Therefore, they are also involved in the schizophrenia biology, synapse formation, neurotransmitter regulation, and many other pathologies involved in developmental process. Astrocytes do play a part. Therefore, under physiological conditions, trophical support of the neuron, as well as the uptaking of the neurotransmitters and deciding in the amount of the transmitter in the synaptic gap, as well as the maturation function of the synapses, and many other functions would be relevant almost all the time to reactive astrocyte function, at least via their moderation. In this respect, I have to say that microglia is another important player. Fully activated microglial cells should also be considered, because under physiological conditions, when everything is going well, microglial cells essentially get into the inflammatory mode and the therapeutic or the repairing mode. These are actually the two ways of activation. If the microglial cells are pathologically activated, then we can speak about a true producer of inflammation markers. Many chemokines, pro-inflammatory cytokines, and excitotoxic products are produced via microglial cells. At the same time, if it's in the reparative mode, it happens the contrary because it would provide a trophic support by way of neurotrophic factors and anti-inflammatory cytokines release. In other words, it would be totally working as a regulatory or repetitive mode. In this respect, we can speak about two distinct ways of activation for the microglial cells. One would be M1 type activation, which is the pathological type of activation, because at the end of the day, the cell is producing or focusing only in the production of the pro-inflammatory cells. The M2 type activation is under physiological conditions, which is most of the time anti-inflammatory cytokine release. In other words, we are speaking about a key cell in charge of balance. In conditions like depression, triggered by conditions like stress, of course, chronic stress would start with the activation of the pituitary axis, that going on with peripheral signals added to neuronal signaling. So it becomes a complicated process. So in that time, with pathological activation, inflammatory microglial cells would come into play in this scenario, where the microglial cells undergo major transformations and suppress neurotrophic signaling most of the time, leading to markers release, and we can name as neurotoxins. This message is transferred to the neuron that it's in close communication with, and in the neuron, stress-linked changes are triggered. Neurogenesis, dendritic branch will be just suppressed, and therefore depression formation due to chronic stress will be triggered as a result of this complicated process. Of course, what I'm trying to summarize here is an inflammatory cascade. Both the immune cells in the periphery, in link to microglia and astrocytes, in linkage to their signals, and the changes in the blood flow to the astrocytes and microglial cells. And sometimes, due to direct traumas or pathologies, we can speak about an influence on the neurons, or again, involving astrocytes and microglial cells. This cascade will be completed with various levels of processes. What I'm trying to tell here is that the immune system and the central nervous system are not very distinct systems that are totally separated from each other. Rather, they are quite linked, and within the central nervous system, as in the peripheral immune system, we do have cells functioning in the same way. And in this respect, astrocytes, oligodendrocytes, microglia all have an interplay with each other, which is quite relevant to the pro-inflammatory cytokine release triggering. In this way, behavioral changes and psychopathologies would be or may be started. Well, if we take a look at the studies in these topics, PubMed screen would lead to the fact that in 1940s, inflammation-related studies were started. Immune and psychiatry, or microglia plus psychiatry terms, key terms entered, revealing really few number of publications, but the real work started in late 80s and continued until today. To appreciate the neurobiology of these diseases better, we need to also look in PubMed. We understand that early 90s studies were started, and quite in a fast way, scientific outputs came up, models validated, and several studies were there. And most of the models, we can speak about microglial activation, relating to depression, which is the essential factor according to these studies as well. Therefore, we can now speak about neuroinflammation as a very debatable matter with a perspective that neuron, astrocyte, and microglia triangle would contribute to the entire triggering of the process, leading to a vicious circle in time, according to all the studies conducted ever since then. Actually, we've seen that soon, and even those people who are not in the scientific environment anymore have learned the term cytokine storm because it became a term they discussed at the news on the news bulletins on TV. After the pandemic, what is important is the effect of SARS-CoV-2. Every cell that I've been speaking about today, which I'm not going to get into the details, contributed to the inflammatory process. All these mediators were produced as a result of this complicated process. Of course, through the respiratory pathway, the virus enters into the body, olfactory nerves, and mediated by the nerves, it would have neuroinflammation in the central nervous system. And then astrocyte cells would contribute to the inflammatory process, strengthening the inflammatory process. And at the same time, peripheric inflammation markers are transported to the brain, contributing to neuroinflammation there. Therefore, we're speaking about a dispersed neuroinflammation presentation. As a matter of fact, it looked quite innocent at the beginning because angiotensin 2 is a molecule that we know with vasoconstriction features. But as a matter of fact, after binding to the ATP, increasing oxidative stress, all the organism is affected, both in the periphery and the central, leading to a cytokine storm, which we never thought before. Both in the COVID process and afterwards, we also tended to see psychiatric diseases, mostly depression, that is considered as a part of the post-COVID syndrome. Essentially, we are speaking about COVID virus, which plays a part in the depression neurobiology. Kynurenine pathway is activated, where the toxic metabolites are significantly increased. Reactive oxygen radicals are just found in abundance, leading to neuroinflammation in the brain, triggering neurooxidative stress in the brain. Today, we know that. To make all of this in a schematic drawing, we see microglial cells. Both pro and anti-inflammatory cytokines are produced, and phagocytosis is controlled as a part of the functions of the cell. Astrocyte can be seen as a part of the regulator at all levels, and many neuroprotective molecules are produced. Activation of phagocytosis is also controlled by astrocytes. And perisynaptic neuron also comes along with these cells in charge of synaptic stability, dendritic growth, synaptogenesis, and neurogenesis. So we can say that perisynaptic neuron is a regulating cell, particularly postsynaptically. It's in charge of the signal transduction mechanism. So there are many receptors and post-receptor events that are taking place in this complicated table. It's a big table. It has many components inside. In the story, particularly recently, there is another mechanism that is added. You would remember, most of you. Physical stress or any kind of signal that would threaten the organism would lead to the natural protective receptors of the body. Patent recognition receptors are activated. These receptors would lead to the macrophage cells and microglia cells on their membranes that are present. Therefore, the receptors that are also stimulated by the stress are our cells that are involved in the activation of the immune system. Once the pattern recognition receptors are activated, node-like receptors will replace these PRRs. Once the node-like receptors are activated in the serum, all kinds of silent cytokines would become from inactive form, converted into the active form, and released. Therefore, inflammasome, which are the intracellular mechanisms, will become the first and strongest pro-inflammatory cytokines, making these cytokines from inactive status to active status. Therefore, it should be considered as a very early phase. I was speaking about toll-like receptors a minute ago. These receptors are present in the membrane of all the cells, represented with many different subtypes. Therefore, I'm not going to get into today, but these subtypes do have many different tasks to support the process. On the other hand, inflammasome are the pro-inflammatory cytokines. Activating the early phase of these, it's not only bacteria or viruses, but at the same time, the ATP, cholesterol, monosodium urate, in other words, sterile activators may also have a part in activating these as well. That is why we need to look into the other parameters, general health condition, as well as the other factors. Inflammasome has different types. Amongst these subtypes, the first one is in the neurons. The second subtype is in astrocytes, NLRP2. And the third one is NLRP3 in microglial cells, which has been looked so far in terms of depression. NLRP3 cells are expressed mostly in the microglia. Therefore, looking at which NLRP is elevated, we may have an idea as to which of these three is more active. We can do that now. With this perspective, we can say that essentially NLRP3 inflammasome would lead to proptosis, neuroinflammation, and dendritic spine losses or ptosis. They are individually involved in these processes, therefore are critical. So the result is cytokine storm or elevation. Psychiatric diseases characterized with cytokine storm or elevation. We can speak about a silent alarm because the molecule remains in the cell before pressing the button. Once the button is triggered, the first pro-inflammatory cytokine release would lead to the activation of the entire process. Stress originating CNS diseases, all of them, almost all of them, as shown by the studies, would lead to inflammasome activation. And inflammasome activation, of course, is characterized by an extreme increase of cytokines. The next speaker will be speaking about nutrition, lifestyle, exercise, how they will contribute to reversal of this process, suppression of NLRP3 activation. So we need to also speak to the, listen to the summary to be provided by the next speaker. So as a matter of fact, if a neuroinflammation is quite active in the pathologies, now that NLRP3 activation is quite important, then the inhibitors of this molecule may be used for therapy. This is the essential third for many of the studies conducted in this respect. This table highlights the relevance of clinical NLRP3 inhibitors. I mean, we don't know, of course, for how long later which of these will be presented, the use of the central nervous system or psychiatric specialization. But obviously, NLRP3 inhibitors are in the center of many of the pharmaceutical research fields. On the other hand, we have a time-related problem, as you would appreciate, ladies and gentlemen, because existing antidepressants and antipsychotics would take a waiting time like 3-4 weeks to get a good response. Actually, this is the group of patients that are not very compliant in pharmaceutical therapies, would have some challenges in regulating the pharmacotherapies, and we expect them to wait for like 1 month or so, even if they wouldn't see a good effect. This is somehow like asking for the impossible. Therefore, we need an early and quick onset action. This is something really needed in the clinical background. Glutamatergic system receptors, namely NMDA receptors, become a really important player here. Ketamine, namely NMDA receptors, both in terms of their different subunits and binding zones, have been of interest to neuroscientists for quite a long time. These receptors, through the ion channel, by way of ketamine binding, will enter inside the intracellular space in controlling the amount of the glutamate amount, and they function as an ion channel. And through this receptor, sodium and calcium entry can be controlled. Therefore, glutamate-related excitotoxicity will be prevented. So the main effect of ketamine would be to prevent excitotoxicity of ketamine, as we considered at the beginning, but in time it was replaced by neurotoxicity, because NMDA receptor blockade eventually contributes to another ionotropic glutamatergic system receptor, AMPA receptors, to mediate the ARCT and mTOR pathways to lead to an increase in dendritic spikes. Therefore, the capacity of the nerves to synapse or the plasticity within hours. So it's a really important effect. The actual fast effect is in the neurotrophic support eventually, as we understand today. Of course, plastic changes inside the central nervous system and the brain would lead to both chemical, structural, and functional changes. The process would be shaped in time. However, what really triggers is the fast-acting agent touches the system as a miracle. Therefore, this is recabling or rewiring of the neuroplasticity. Therefore, this new structure contributes to a faster communication and between the neurons and reshaping the system. Of course, here neurotrophic factors and the trophic support, as I said, for the microglial cells is a key step, because neurotrophic factors such as BDNF are found on the surface of these cells, presented with TRKB receptors for BDNF, because as you know, these receptors and neurotrophic factors are quite variant. The produced BDNF are bound to TRKB receptors. Dendritic elongation contribute to synaptic capacity, which is a really important mechanism to play in this process. Thanks to that process, once the activation is triggered once, then we would see many signal transduction mechanisms inside the cell, each of which, of course, are quite detailed to be unique topics of talk, but ARC-mediated growth, ARC-mediated survival, and phospholipase C-mediated messenger system would also contribute to synaptic plasticity. That's the only message I'm trying to focus here, because the increase in the production of BDNF, I think, is quite relevant as a first chain to trigger the entire process. This study is quite interesting, published in the Cell in 2021. What was surprising about the study was that the RKB receptor we used to know is that the binding place But now the receptor is quite active and the presynaptic neuron is active. Only then ketamine-like antidepressant agents also can be used for the antidepressant effect, and the clinical practice can also be bound here. Here, the neuron cell should be live and active, provided that some antidepressants can bind to that location as well. Maybe this can be a new horizon for that aspect. I have been speaking about sensitive balance since the beginning of my talk. Pro and anti-inflammatory cytokine balance must be preserved for this. Of course, sensitive balance, they are produced in our body all the time. If the balance is impaired on the favor of any of these, everything would be devastated, triggering a diverse and a widespread inflammation cascade. So in this talk, I have been talking about neuroinflammation in psychiatry and the crosstalk between CNS cells. But it's like a snapshot. I tried to open up a window for you in the neurobiology of psychiatric diseases, such as depression. May also be contributed from an increase of neuroinflammation, neuroplasticity, and also glial cells, microglia. Astrocytes, oligodendrocyte cells do have a direct contribution to the process, as well as neuroplasticity. Early pro-inflammatory cytokine release will have a contribution of inflammasome. And the neurotrophic factors are triggered by mTOR phosphorylation. In this talk, I tried to summarize all these aspects. Well, how about all these mechanisms? Will they end up with drugs in the pipeline? And can we use all this information? The last 3 slides I have, very quickly, I will summarize, and I will try to show you whether there is a drug effect of all this knowledge. Characters are not appearing properly on the screen, but hopefully you can read it. Potential indication is bipolar depression, oral route, fast-acting antidepressant class. So this molecule is produced for this intention. Actually, antipsychotic lurasidone, and the cycloserine combination. This preparate. And the hypothesis is NMDA receptor blockade results in glutamine levels, particularly on the anterior cingulate cortex, with a particular regulation there. It's in the phase 3 clinical trial level. The second molecule we need to look into is hydroxynorketamine. It is for major depressive disorder, and this study is run by NIMH. Here, hydroxynorketamine is the active metabolite of esketamine and arketamine. As I have just spoken in my talk, mTOR mechanism is to be used to support the synaptic plasticity, according to this study, phase 1. And the last one is the NV5138 molecule to be administered in the oral route for major depression. Again, mTOR mechanism is to be the mediation. So eventually we can say that the future therapies are quite promising. Based on the new results we're obtaining in neurobiology, things will be redesigned, hopefully in the best possible way, more optimistic way. But the take-home message is, and which was the topic of my talk, neuroinflammation and neuron microglia crosstalk is extremely important. Together with the triggering of the astrocyte cells, this inflammatory process is somehow like an on and off button working in the brain. And the true remission can only involve these mechanisms with the drug molecules we are going to design in the future. Thank you very much for your attention. Hopefully it's been a thorough summary without boring you. Thank you.